Hello everyone, welcome to this new series about 100 most important topics for your prelims 2026. And again, today this video is dedicated towards the science and technology portion, and we are talking about very important perspective. And in this video we are going to talk about the fundamental of your genetics, that is very important. So fundamental of genetics, DNA and RNA, basically we are talking about the basic structure of it. But the most important thing, that is your transcription, translation and chromosomal inheritance, because this thing is very much important for knowing the genetic engineering, or maybe we can say genomic engineering, where the most hottest topic is your CRISPR-Cas9 and base editing and the Genome India project that is very highlighted in current days. And also we are talking about the assisted reproductive technology, that is called your ART, where the 3-parent babies are there, mitochondrial replacement therapy is there, and IVF breakthrough is there. So this is the content that we are going to talk about in this video. So in a very first slide, we are going to talk about the basic structure of DNA. I know you know it very well, but this is the one of the most important basic information, that is called your DNA, because DNA in our body carry the basic information, genetic information of the body, and DNA is very complex, which is related to the all character of animal, plant and human also. So when we talk about in a very basic manner, so you can see there is a use of deoxyribose sugar. So we are using the deoxyribose sugar for making the DNA, and also there is some bases are used. So there is adenine, guanine, cytosine, and actually this statement is little bit wrong here. So we are using the adenine, guanine, cytosine and thymine for DNA. So this is your 4 type of nucleotides that we have. And you know that when we talk about the adenine, guanine, cytosine and thymine, we called it nucleotide structure, and these nucleotide is of 4 type, but we can divide it into 2 part. The first one is known as your purine bases. These are known as your purine nucleotide, where the adenine and guanine is present. And when we talk about the pyrimidine, so this one have guanine and thymine. So these 4 type of nucleotides are there. And also when we talk about its structure, so it is a double helical structure. You know that Watson and Crick are the scientists who can give you the structure of double helical. And there is a anti-parallel structure, means they are coiled and they are basically rotating in opposite direction. And also right-handed helix is there. This structure is given by Watson and Crick model, you know it. And there is a base pairing, because we can connect the thread, like you can see in this DNA structure, you can connect the 2 thread of DNA by making some bond, and these bonds are present in between the base pairing. So there is a adenine always connected with thymine. The most important word, there is a 2 hydrogen bonds are present. But when we connect guanine with cytosine, there is a 3 hydrogen bonds are connected, which is more stable, because you know that triple bond is more stable than the double bond. But this one is very important. In DNA structure, if there is a in one side, then T is always in the other side. And as the same way, if there is a cytosine, so in the other way, there is a guanine is present. When we talk about the function and stability, so function is storage for transmission, hereditary information, and stability is very high. So the stability of DNA is very high. When we talk about the comparison between stability of DNA and RNA, so we find that RNA is something which is present in older time and then DNA is made, but DNA having a hereditary information more stably, and that's why in this universe every living being have the genetic information in DNA. So genetic material in every living organism, although it can be plant, it can be animal, or it can be any microorganism. So all have that DNA, but only viruses, which is primitive in nature. So viruses have RNA as a genetic material. That is for the exception. Rest thing, when we talk about where this DNA can be present, so you know that DNA is present in both eukaryotic and prokaryotic cells. fully developed cell eukaryotic, undeveloped, less developed cell is prokaryotic. And this eukaryotic cell have it in nucleus, mitochondria and chloroplast. Sometime this kind of the question is also asked in UPSC. Already UPSC can ask this question, which part of cell have the DNA? So there is a mitochondria, chloroplast and nucleus. But the main DNA is present only in nucleus, and both this part have extra DNA. Both this part have extra DNA. But when we talk about the prokaryotic cell, there is not a complete nucleus is made. So it is present in nucleoid region, means nucleus like structure is there where the DNA is present. So this is something that is called your DNA. And when we compare it with your RNA, so basically RNA is a half structure of DNA. Like you can separate the two thread, and one half thread is basically known as your RNA. So RNA is basically a single thread structure. You can see, you can half this structure, and this half structure is your RNA, ribosome, ribonucleic acid. When we talk about the sugar, so there is the ribose sugar. DNA have deoxyribose, this one have ribose. There is also base pair, but adenine, guanine and cytosine, and now you have uracil in the replacement of thymine. So that is the major difference between DNA and RNA. So the first difference, DNA is double helical, RNA is single helical. Second one, the difference, sugar, deoxyribose in DNA, ribose in RNA, and also the change in base pair. Adenine, guanine, cytosine is same in DNA and RNA, but on the replacement of thymine, uracil is present in RNA. This is the major change. This structure is usually single stranded, and there is a three type which is very important. The first one is known as mRNA, M which carry the genetic information, so we called it messenger RNA. What we called it? We called it messenger RNA. It can carry the genetic code. tRNA, which brings the amino acid, and this one activate when we want to make the protein inside the cell. So this protein information is given by mRNA, but for making protein, amino acid is carried by tRNA. And the factory of our body protein is ribosome, and this ribosome can be activated by your rRNA, which form your ribosomes of the body. Basically, this thing is very important. We also look at this part, how it can be functioned. So as I tell you, DNA have all genetic information, and you want to make protein in the body for your growth. So mRNA carries the message, tRNA transfer amino acid, and ribosome is given by rRNA. So your body cells can make proteins. This is known as your central dogma of biology. Clear? Next one is function. So basic function is protein synthesis, because RNA is a message for protein synthesis and its regulation. And we know that it is less stable than DNA, because RNA comes first in the nature, then it formed DNA. So DNA is much better, much stable than RNA. So DNA is more stable than your RNA. So this is known as your structure, half helical. Now we talk about the central dogma. This is a very big concept. Like every living being have a simple rule, and this rule is every cell of the body have DNA, which have all genetic information, but the result of every cell is protein. Every cell want to make protein, but DNA cannot directly make protein. First, DNA give it in the form of information, and this information is given in the form of RNA. So first DNA can form RNA, and this RNA give this information to ribosome, which is basically known as your factory of protein. And now the proteins is made in your cell, and you can see this all process is controlled by some enzymes. Every living being have simple this process, and this is known as the basic life process. That's why this is known as your central dogma. It means it is present in every living being. Like you can see the structure. So you find this is your DNA, And when DNA make its copy, there is a replication. What is the process? Replication is happen. So replication means making copies of DNA, and when this DNA send a message for protein formation, so this is known as your translation. What we called it? Translation and transcription process are there. So when DNA is giving the information in the form of RNA, this is known as your transcription. And when protein is made by ribosome, this is called as translation. So there is a three process. The first one is replication, making copies of DNA, transcription, making RNA from DNA, which is basically a information to ribosome. And when ribosome make the protein, this is called translation. Lation word define the synthesis of protein. And you know that when we talk about making RNA from DNA, this one is controlled by RNA polymerase. This is a very important enzyme. So RNA polymerase is the enzyme which can helping in making RNA, and then it can go to this ribosome, and this ribosome can make different type of proteins in the body. So this is your central dogma, and you can see here the involvement of mRNA is there, because mRNA is the structure which provide the message like which kind of the protein is made in body. And this is the process how our body can change its function in different area. Like our hair cells can make the keratin protein, but our muscles can make actin and myosin protein, but our bone can make osin protein. So different type of protein making, this process is fully controlled by DNA, because DNA have all information, but it can first give to RNA. RNA is the message, mRNA is the message. This one reach to ribosome, then ribosome can form the proteins. Clear? But there is one process which is very important also, and this is called as reverse transcription. So you know that transcription means there is a formation of DNA to RNA, but if you can reverse it, there is a RNA making DNA. So you can see this is the viral RNA. So when a virus can be entered into your body and they have the RNA, so now you can see this RNA, viral RNA, can reach to your cell DNA. There is a cDNA word. cDNA means this is cell DNA, and cell DNA have this viral RNA which can overcome the structure of your body function. The cell can be captured by your virus, and then there is an integration of cellular structure with it, and then new DNA is formed. So double helical structure is formed. So single helical RNA form the double helical DNA. This one is reverse in nature. That's why we called it reverse transcription. And this kind of the function can be happen when our body can have viral infection. So when virus can reach to our body cell can be captured by virus, then it is used for making its whole stable structure. And that's why we can say viral disease can get more stable in the body and they can cause very harmness in the body. So this is known as your reverse transcription. Now we also go to talk about some chromosomal inheritance. This is a very basic information related to your genetic chromosomal information. So chromosomes is basically your structure which can pass through with the parent to our body, and we also based on that structure that we can donate it for the future generation. So chromosomal inheritance means that genes are carried on chromosomes. Genes means the characteristic of the body, and these traits means characters are passes from parent to the offsprings through these chromosomes. The important principles behind it, there is a genes are on the chromosome. This is a very informative thing. Genes are located on chromosome, also located on DNA. Actually, the coiled structure of DNA is known as chromosome. So the genes characteristic part are present on the chromosome, and each chromosome carries many genes. So genes have a fixed position on it, means there is a fixed loci. There is second thing, chromosomal occurs in always pairs, and we know it, human body have 23 pair of chromosomes. So they are always present in pair. So there is 23 pair, 22 pairs are autosomes. Autosomes are those chromosomes which can be similar in both male and female body. But there is a one change being male and being female, that is use of one sex chromosome. And you all guys know that if 23rd pair of chromosome have XY nature, then the body is male, and if the combination is XX, then body is female. This is the basic thing related with chromosome. Now we talk about the segregation during meiosis, and you might be know about it. Meiosis is the type of cell division which can done only in sex cell, or maybe we can say sexual cells, for the production of sperm in male and for the production of ovum in female. So during that process in meiosis, there is a homologous chromosomes are separated, means chromosomes can be separated into half structure. And you all guys know that parents can give half DNA to the next generation, like father can give the half DNA and mother can give the half DNA by their own sperm and ovum, zygotic cells. So this is basically known as your segregation. So they form the gametes, and gamete means sperm and ovum. Sperm and ovum. This one explains the Mendelian law, that is called as Mendel's law of segregation. Then there is also a rule that is called independent assortment. Different chromosome pairs separate independently. It means one chromosome never be affect the function of second chromosome, or maybe we can say every gene have its own specific function. This is independent assortment. And the most important process is crossing over. I can make it very simple for you. Like you can observe, a parent have two babies. One, both can be male, both can be female. Maybe one is male, one is female. But if there is a two boys are there, two girls are there in a same family, so both cannot have same body character, but the parents is same. So when each and every time parents can donate its sperm and ovum, means half of the DNA, the structure and combination is changed. So every time this combination is changed, and this combination is known as crossing over. So we can say the exchange of genetic material between homologous chromosome, it is occurs during the prophase 1 of meiosis, and basically prophase 1 is the phase of cell division where the chromosomes getting separation, and it create the variation. That is the point that I tell you in a simple manner, that same parents have different body structure babies. Just because of that, every time the variety can be changed. This is called crossing over. When we talk about the last one, you know it, there is a sex linked inheritance, and this one is directly related to 23rd pair of your chromosome. 23rd pair of chromosome. So genes are located on X and Y, and you know that this one is made by X and Y. And there is a disease which is directly related to X. So X-linked disease are there, like hemophilia and color blindness. And due to X-link, you know that male body have only one X, female body have two X. So this disease can defect in X. So male have more effect on it, but female does not have as much effect as male have. So we can say this disease can be more dangerous for male and less for the female. So mostly they are related with X. So the more common in males. This is the simple thing. And now we are entering towards the genetic engineering, or maybe we can say gene editing therapies. And this is your latest technology that is called as CRISPR-Cas9 technology. And you know that in the biotechnology section, when we talk about required change in genes, or maybe we can say biotech provide you the base for changing the structure of gene and DNA. So there is one most important technology that is called CRISPR-Cas9 technology, and the word called CRISPR. CRISPR is basically clustered, regularly interspaced, short palindromic repeats. This can represent you the structure of DNA, the full form of CRISPR, but the most important thing is there, that is Cas9. And Cas9 is a endonuclease enzyme. It means it can be cut the DNA from any part of the midpoint, and that cut the DNA at specific target sites. And it uses the guide RNA because we have to send a message that this is the area, or maybe we can say this is the defective part of your DNA that you have to be cut. So for the cutting location precisely, we give it a guide RNA that is called gRNA to identify the target DNA sequence. What is the advantage? Because if you can give a message for precisely cutting, so the advantage is high precision, low cost, and faster than earlier methods. When we talk about the application, so we use it for gene therapy. And you know that gene therapy is the treatment of genetic disease. Genetic disease. So nowadays we can think about the process like genetic disease can be treated by gene therapy, where precisely we can cut the defected gene from any human body. There is also genetically modified crops, like you know that we change multiple type of crops by this process. And there is a disease research like cancer and sickle cell anemia is there. So this is very important one. Next, and the new process is now very talkable. This is called base editing. It is almost similar to your CRISPR-Cas9, but little bit change are there. And what is the difference? It is an advanced form of your genome editing. Advanced form, next generation CRISPR we called it, and it can use also the Cas protein for cutting the DNA. So what is the advantage over the CRISPR-Cas9? What is the benefit? So reduce unwanted mutation is there. So uninvented mutation cannot be happen with this condition, and it is higher precision than your CRISPR-Cas9. What are the application? So it is the application for correction of point mutation, and point mutation means your DNA can be changed its structure in only one point, which is very short in nature. So you can do a very precise method, that is your base editing. And the treatment of genetic disease, this can be common in both base editing and CRISPR-Cas9. Next one, if you compare the basic features of CRISPR-Cas9 and base editing, this one is very important for your prelims. When we talk about DNA break, there is yes, it can break the DNA, but base editing cannot break the DNA. Precision is high, but much higher in base editing. Mutation risk, this is higher in this part, but lower in this part. As you see, this is the major difference. This is suitable for insertion and deletion. Insertions means if you can put a new gene on it, and deletion means if you only cut the gene from it. So it is used in both, but this one is for point mutation. That's why it is called more precisely method than CRISPR-Cas9. If we talk about the next one, this is our indigenous technology, like Indian scientists can develop this part, and this one make CRISPR-Cas9 better than the older procedure, because during this procedure there is a lightning effect is come, because we can use a bioluminescence animal gene with it, like we can take it from prawn body that is present in deep sea in a dark area. So we use it a gene which is making a light on precisely cutting for DNA. So when you use the Cas9 protein for cutting the gene or DNA, there is a light illumination can be come out. This one help you for the better control and better visualization over it. So this is known as your CRISPR Glo9, very important one. So GloCas9 is a modified version of your CRISPR-Cas9 genome editing tool, which Cas9 protein tagged into a fluorescent marker, means producing light on it. The glow word refers to the fluorescent signal that allows scientists to visually track the Cas9 activity inside the living cell. So this one make it more precise. How it work? Cas9 enzyme is fused with a fluorescent protein. This one is taken from prawn. This one is taken from prawn, and the guide RNA directs Cas9 to the target DNA sequence. So target DNA sequence is changed and illumination is there, Cas9 glow, showing where and how editing can be occur. The researchers correlate glow intensity with the editing efficiency and safety. So this one make this cutting procedure of any gene and DNA is very great. So this one is very important procedure. And when we talk about the difference, this is again very important for your prelims. When we talk about conventional Cas9 means older one and your glow Cas9 which is the newer one. So visibility is not present in conventional, but now you have the fluorescent light visibility. There is a tracking also there, but that is indirect. We track the result of genome editing, but now we can check it on real time, because in this part first we done the editing, then we can see the result. In this part we can precisely control the real time editing. When we talk about the UPSC relevance of it, that is very important. What is the core concept behind it and advanced application is that. So this is your core concept for cutting the gene, but this one make you very advanced in application part. Now we talk about the Genome India project. As you know that when America can done their GIP, then after that we also want to study the Indian genome. This one is very important. So we start our program that is called Genome India project. This is a national initiative for mapping the genetic diversity in India, because in this part we are taking 10,000 plus Indian samples, means we are taking 10,000 plus Indian individual samples, and this is led by your biotechnology department, Government of India. There is an institution, ISC Bangalore, IITs and CISIR lab. What are the objective? So we create a reference Indian genome, because when America can done their genome project, so there is a basically lots of their people are there, but for Indian perspective we need our Indian genome. So we are using this Genome India project. Understand the population specific disease, why some disease can affect same kind of the population, and we also promote the precision medicine, because after the studying of a genome of any person, we can design the medicine particularly for the affected person. And this is called as your modified medicine, or maybe we can say special characterized, special create medicine, which can very effective for the affected person because they can design it. So this is commercial medicine precision. And what are the significance for it? So help in personalization of healthcare, as I tell you, personalized medicine, and then strengthen the Indian bioeconomy, because this one is very helpful for many type of researches. This is known as your ART, assisted reproductive technology, and this is very talkable in partial time, but now we have to study about the concept of it. So this is a medical procedure used to treat the infertility. ART is basically helping type of technology for the infertile couples who want the baby, but the body cannot be enough capable for getting it. So it is a medical procedure used for treating the infertility and handling the gamete. And it includes some kind of the basic thing, IVF, which is known as your test tube baby, in vitro fertilization, there is ICSI, intracytoplasmic sperm injection, how you can enter the sperm into womb. Gamete donation is there, like sperm donation, gamete donation. Embryo transfer is there, like you can transfer the full embryo with it. And cryopreservation, freezing of gametes, means sperm and ovum. This is newly in India, sperm and ovum. This is known as your ART. When we talk about its effect, so the first and most important part is your IVF. So fertilization of ovum and sperm outside of the body, means outside of the mother body, and you can just implant the embryo into the uterus. What are the steps? First, you can stimulate the ovarian stimulation, egg retrieval, means you can take the egg from mother body, then fertilization in lab, embryo culture, you can take it for 7 to 8 days for growing the embryo, and then there is a embryo transfer is there. What is the recent thing? We find the first thing that single sperm injected directly into womb, PGT is there, like pre-implantation genetic testing, means you can change also the genetic of baby. This one actually be very prohibited thing, but detected genetic disorder. Like why we can prohibit it? Because the people can misuse it. But this one is a very precisely technique where you found that the upcoming baby cannot have any kind of the genetic disorder. There is a time lapse, embryo imaging, better embryo selection. These all things are there, which is now included in India. Like in current days, you can choose the sperm by seeing the features of the body, and also you can choose the ovum by seeing the features of the body. So basically, current days, IVF is something bigger than what we want to know about this thing, that sperms are donated with their feature. So some people can donate their sperm, and someone who need it, they can just purchase it by making the structure of baby. Like a person is very tall, fair in skin, so they can give the characteristic about this, and you can just purchase it for your upcoming baby. This is the new things related to IVF. But when we talk about the cryopreservation, this is the new thing. As I tell you already, cryopreservation means minimum temperature preservation. So you can preserve the ovum and sperms. So long-term storage with highly survival. IVF does not alter the nuclear DNA. This is the main thing which is important. That's why it is allowed. And unless combination with the genetic technique. Like as I tell you, we cannot change the structure of a baby, but one couple can select the structure of ovum and select the structure of sperm. So the baby can be designed, but in a natural way, not by the biotech processes. This can be used for infertility and genetic disease prevention. Now, this is a very interesting topic that is called 3-parent baby. Yes, that can be possible. So this is the baby with genetic material from 3 individual, like nuclear DNA. Nuclear DNA means main DNA. How does the body characteristics are made? Nuclear DNA, biological mother and father. So there is some couple where mother cannot be capable to produce the baby, or maybe we can say there is an issue with mother mitochondrial DNA. Like you can simply say, maybe a mother have a genetic effect, but she is only the vector of genetic disease. So she is not affected by the disease, but upcoming generation can be affected. So basically, both parents donate its own sperm and ovum for fertilization, main nuclear DNA, but the defected mitochondria is replaced by a donor female. Because the mother can give a defected mitochondria, so we replace it. So there is a basically 3 persons are involved. That's why we called it 3-parental baby. The purpose is to prevent the mitochondrial disease inheritance, because maybe the actual mother can feel she is a vector for any genetic disease. So we prevent it by using the mitochondrial DNA from donor female. What is the country status? So it is legal, permitted only in the few countries like UK, not permitted in India. It is yet not permitted in India, because you know that Indian people can use it in different manner, so we cannot use it rightly. This one is also known as mitochondrial replacement therapy. As you see, in 3-parent baby, we replace the defected mitochondria by healthy mitochondria, and this is your mitochondrial replacement therapy. So a reproductive technique to replace the defective mitochondria in mother and egg with healthy mitochondria from donor egg. So we know it. What is the technique? Maternal spindle transfer is there, nuclear DNA transfer before fertilization, and pronuclear transfer is there, nuclear DNA transfer after the fertilization. So there is a 2 process. This one is very important. First one is nuclear DNA transfer before the fertilization, and maybe you can transfer it after the fertilization because you just want to change the mitochondria. So before the fertilization it can be happen, after the fertilization it can be happen. What is the benefit? So it prevents the disease like syndromes, mitochondrial myopathy, and key fact, MRT modified mitochondrial DNA, not the nuclear DNA. This is the reason why we accept it, and MRT is not a cloning, because cloning is fully banned in India and world. Next, ethical concerns, scientific concerns are there. So there is a germline modification, means body structure modification, not the nuclear. Identify the consent of donor, long-term health impact, which is unknown, and slippery slope towards the designer babies. So we can say this kind of the concerns are there, because that's why India cannot use it rightly, because the people can design their baby with this process. So we cannot allow right now in India, but some countries like UK can allow it. And now we talk about surrogacy. You know that after 2002, India is a big market for surrogacy, about $500 billion market. But when we see our females exploitation are there, so we ban it completely. And in India, the foreign person cannot take the surrogacy, and we are limitize this surrogacy facility totally. So after 2012, we can ban it, and now we have a proper law for it also. We talk about it. So surrogacy is simply a woman carries a child for another person and couple and hand over the baby after birth. So surrogacy is basically we can take a mother for giving the birth of a child. Two type of surrogacy is there, gestational and traditional. Surrogacy has no genetic link, and when surrogate mother egg is used, so it cannot allowed in India. So we have one thing that only we are using the body of something other female for delivering the baby, but we cannot use the biological egg or any DNA of it. So in India it cannot be allowed, only gestational is allowed. When we talk about the Surrogacy Regulation Act, we have a proper act for it, because as I tell you, female exploitation is done in India. So only allosteric surrogacy, no mandatory compensation, like no money exchange is there. This is happen only for the married Indian couple, which proven its infertility. So when the couple can prove we are infertile, then it can be only allowed in India. It is prohibited. What is prohibited with this law? Commercial surrogacy, means money exchange for it. No. Foreigners, live-in couple, same-sex couples, and single men, they cannot capable to take the surrogacy services. And the surrogate mother, there is a rule for surrogate mother. She can be married, have at least one biological child. The age is between 25 to 35 and can be surrogate only once, because this one is only done only for the help for infertile couples. What are the authorities which can have a take control over it? This one is National State Surrogacy Board and Registration of Clinical Mandatory. This can be your Surrogacy Regulation Act 2021. Talk about ART. So there is Assisted Reproductive Technology Regulation is also there. And when we talk about the ART thing, it is key difference from Surrogacy Act. It covers the IVF clinics, ART banks are there. It allows the single woman to use the ART. Single woman can use the ART techniques. There is a gamete donation, embryo storage, and clinic ethics are standards. So this is the regulators. What are the penalties? Sex selection is completely prohibited, because this is the problem with this technology. The people can use it badly. Violation, heavy fines and imprisonment. What are the facts related with it? Mitochondrial DNA is maternally inherited, means mitochondrial DNA can become from mother. 3 parent babies involve mitochondrial replacement, not cloning, because cloning is banned. Only mitochondrial replacement is done. India allow allosteric surrogacy only, not for the commercial surrogacy. ART Act and Surrogacy Act were passed in 2021, and MRT effect less than 1% of the total DNA. So it is very rare for the couple, but it have to be as after 2021. So these are some questions for you for your practice. So I put the 3 questions after this video for your practice, and you can watch this video, clear your concept and give the answer in the comment box after this video. So thank you for watching.
For more informative content, like, share and subscribe, and do not forget to press the bell icon to get the notifications.